Hello, my name is Matteo Fratarcangeli, and this summer I teamed up with Dr. De Silva to do a research project focused on studying the luminescent properties of europium metal-based nanoprobes for potential biomedical imaging. Current cancer imaging technologies rely on intricate instruments, require high-power lasers or radioactive probes to detect cancer cells. Because of these, a patient can find himself paying thousands of dollars in the first years of treatment. Development of luminescent particles based on europium metal could reinvent the way the cancer cell is diagnosed in the near future, as it would allow a doctor to image cancer cell using low power and low cost radiations, such as UV, infrared, and visible light. An example can be found in Figure 1, where UV light is used to illuminate the cell in green and the probe in red. In this project, we studied the molecular models of the nanoprobes created with a computer software to improve the glowing intensity of such probes. Using only europium metal ions was not optimal because the glowing intensity is very low. So we chemically combined europium metal ions with organic molecule called ligands to increase the luminescent brightness. Figure 2b is a prototype I made in the lab before the lockdown. And if we were to zoom in, we will be able to see the molecular structure of the probe represented in Figure 3. In this model, we can see the europium ion on the right in green, responsible for the glowing, and on the left, the ligand, which is responsible for the intensity of the glow. This is also the part of the probe that can be modified to improve the glowing. Our focus last summer was to study the electronic energy levels of the nanoprobes so that the particle with higher luminescence can be identified, thus helping with the design of highly luminescent cancer imaging nanoprobes. To do so, we used computer simulations and calculations that provided us with the data we needed. We built 9 different ligands with small structural changes and ran calculations to define the structure and obtain their electronic energy levels. It turns out that each ligand has its own unique energy level. We have determined optimal energy levels for intense glowing. As a result, energy levels that are higher or lower than the optimal ones correspond to decreased glowing intensity and to a suboptimal nanoprobe. In conclusion, we were able to correlate the luminescent efficiency of the nanoprobes with the electronic energy levels of the ligands. Our findings can be used in the design process of luminescent nanoprobes to predict how bright the probe will be. Future work includes simulating nanoprobes with different ligands and making the studied particles in our chemistry lab to carry out preliminary imaging studies using live human cells.